Welcome to a special Wednesday afternoon lecture, the Florence S. Mahoney Lecture on Aging. Some of you who have not been around as long as I have might not know who Florence Mahoney is. Well, she was a legendary mover and shaker that had a lot to do with attention being paid to biomedical research going back a few decades. She and Mary Lasker, a name that you may know better, were close buddies. And the two of them were absolutely incredibly effective in getting attention to the importance of supporting medical research and having the government be a major supporter of that. Jack Valenti, who was a friend of Mrs. Mahoney and was a special assistant to Lyndon Johnson, tells this story that when he would tell President Johnson that Mary Lasker and Florence Mahoney wanted to see him, President Johnson would groan. He'd say, "Oh my God, these two women are going to bankrupt the country." And they usually got what they wanted from President Johnson and others as well. And so, not only did she support NIH in general, she had a particular interest in aging, and the creation of the National Institute of Aging in 1974 was something that Florence Mahoney strongly supported and probably had more of an effect in making it happen than anyone else. She also continued to be very interested in NIH's goings-on until her death at 103, and that was back in 2002. But Richard Hodis tells me that before that, and right up to very near the end of her life, she wanted to have personal input into who gave the Mahoney Lecture. So Florence, if you're up there, I hope you're happy to see that today's choice is a distinguished scientist who has contributed significantly to our understanding of the molecular basis of senescence and aging, and happens also, by the way, to be the director of the National Cancer Institute. Ned Sharpless, arriving here as the director of the NCI, appointed by the president, began his undergraduate career at the University of North Carolina as a Moorhead Scholar, stayed there to do his MD, a place that I think gives pretty good MDs since I have one of those myself. And after training at Mass General and the Dana-Farber and a couple of years on the faculty at Harvard, went back to the University of North Carolina in 2002, where he remained until last year. He was appointed to come here as our director of the NCI. He has done distinguished research, particularly focused on a molecule that I'm sure you're going to hear more about called P16. P16 has this wonderful attribute of being a way in which we are understanding the connections between aging and cancer, probably more than any other such molecule. And I guess it's highly appropriate, therefore, that Ned represents also that intersection, having done himself remarkable primary work in the biology of aging, and now as the director of the National Cancer Institute, spreading across those presumably rather porous boundaries in a very effective way that you're going to hear more about. He has a provocative title, Senescence, Live Fast, Die Young, oh yeah, and leave a good-looking corpse? Well, that's what he said, so let's hear what that's all about. Please join me in welcoming Dr. Ned Sharpless.
Thank you, Francis, and I'd like to thank the NIA and Richard Hodis for inviting me to give the Mahoney Lecture today. I think, since arriving at the NIH, I've really enjoyed the interactions among the different ICs and the federated nature of the NIH, and it's a real strength of the institution that, I get to work with such great scientists across the entire enterprise. So I thought I'd talk today a bit about work that for the last sort of 2 decades that my lab has done on the relationship between cancer and aging. The way I got into this as an oncologist, I was sort of trying to study the earliest features of malignant conversion and what prevented that from happening, a bunch of tumor-suppressor mechanisms involved in that. And it turned out that some of those tumor-suppressor mechanisms that were important to not getting cancer we discovered to be age-promoting, and I'll go through that in rapid fashion. First, I'd like to, in abundance of caution, as I entered federal service, I divested all worldly possessions, as you would expect. However, I did have commercial relations in the past that are relevant to today's talk, so G1 Therapeutics is this company that makes CDK4 inhibitors. I'll talk a little bit about that. And HealthSpan Diagnostics is this company that measures aging with P16, and I'll talk a little bit about that. And Unity Biotechnology is this company that does Senolytics, and I'll talk a little bit about that too. So there's a lot of interesting things about cancer and aging. You can take this talk many different directions. For many years, it's here, isn't it? Let's see. Yeah, for many years, the top part is really what my lab focused on, is sort of how cancer causes, or resistance to cancer causes some aspects of aging, and how the why there's this strong association between aging and cancer. And that turns out to be a number of interesting things, like genomic instability induced by telomere dysfunction is a big driver of cancer, we think. There's this sort of stem cell aging that I will talk a bit about that represents a change in epigenetic state and dysfunction of stem cells, and then somatic mutations in stem cells that accumulate with aging. And this interesting topic of clonal hematopoiesis that I think is fascinating. There's loss of cellular immunity, which is probably important in monitoring for would-be cancer cells and censoring them, and a pro-tumorigenic milieu that occurs with aging that's quite interesting. And then also the bottom here is a really interesting topic that I think is of great importance in the area of geriatric oncology now. How the fact that most patients with advanced cancer in the United States are over the age of 65, at some point the age of the patient will limit the ability to treat the patient. So there's interesting interaction between a patient's physiologic age and their ability to be cured by cancer therapy. And that's a really interesting topic that I think is emerging in geriatric oncology, and I think probably deserves more attention, particularly this idea of resiliency. You know, if you have a patient who looks pretty good and you treat them with aggressive therapy and then they never recover, how is that patient different from a similar aged individual who gets treated with aggressive therapy and then is back playing golf 2 weeks after they finish therapy as if nothing ever happened? So why some patients who are age 70 bounce back and others do not is poorly understood, but it is biologically encoded in the individual. I think it should be a knowable thing. So, but I'm not going to talk about one or three here. I'm going to really focus on the middle one today about this idea that cancer therapy is related to aging. I think it builds on a modern understanding of aging in a variety of ways. And I've come to call this topic iatrogenic aging, or physician-induced aging, and it relates strongly to cancer therapy, and not just cancer therapy, but we'll talk mostly about cancer today. So that's, and I thought to make one point explicitly, I think a problem with aging talk sometimes is that we often focus on the one part of it we're interested in, and thereby provide the incorrect impression that aging is a simple process. It is a very complicated thing, and different tissues age in different ways, and the molecular basis of aging is quite different from tissue to tissue and even between individuals. And so I think it's important to state up front that I will talk about two kinds of aging that are of interest to me. One is this accumulation of senescent cells, the so-called senescence theory of aging, and the other is this change in stem cell function that is not senescent in basis, that is not senescence, but is a different kind of thing. But I don't think that is a complete list of the mechanisms of aging in individuals, and I think that these are two ways that are relevant to iatrogenic aging, but I think that there are other kinds of aging that are important as well that are not these two things. So let me start with senescent cells. So this is this idea that when cells become damaged, they somehow know that they're damaged, and they activate a program named cellular senescence, discovered in the 1950s by Leonard Hayflick. And these cells may get cleared at some rate, but often they don't. And then we believe they hang around for a long time in an affected individual. And the good news is that's an important anti-tumor break. So this damage often is of the DNA. It's the kind of damage that becomes fixed with proliferation and would be the substrate for malignant conversion. And so a cell somehow knows it's headed down that track and stops doing that through activation of p53 and p16, two of the most important breaks on senescence. So that's the good news. You don't get cancer. But the bad news is we think those cells can hang around for a very long time, and they do stuff that's undesirable, and they contribute to the pathology associated with advanced age. And one real problem in the field has been identifying senescent cells. So there still really is no good single marker that one can do for senescent cells in vivo. But I use this combination of, generally they make p16. That turns out to be kind of the best marker of senescence in vivo. They don't divide. That's like a sine qua non of a senescent cell, is it's non-replicative. And then they have some other stuff, but the one that turns out to be sort of most generally used is they make these factors, these cytokines called the senescence-associated secretory phenotype, or the SASP. And so sort of the working definition in my lab has always been a non-dividing cell that makes P16, that makes some elements of the SASP. That is a definition that many people in the senescence field think is too loose and prefer a more rigorous, they've got to have DNA scars or they've got to have short telomeres or things like that. But I think that definition has the benefit of being like actually usable in the real world, whereas many of the more precise definitions are solely limited to in vitro studies. But so they have a number of these features, and they can be demonstrated in many tissues with aging. And I think for many years it was argued about as to whether or not these were an in vitro artifact. I think now the evidence that senescent cells occur in vivo with aging is reasonably strong, and I won't go through that today. But still, their identification in any specific tissue in a unique individual can be challenging. And so I like to show this slide. This is when Dr. Hayflick discovered senescence. He was trying to actually grow viruses in primary cells. He didn't want to use transformed cells like HeLa cells because he worried they might already have some virus. So he discovered that primary cells, fetal lung fibroblasts, would stop dividing with very reproducible kinetics in vitro. And he tried to publish this. He thought this is something interesting and unusual. So he sent his paper off to the Journal of Experimental Medicine in 1961 describing senescence. And it was rejected because the largest fact to have come out of tissue culture in the last 50 years is that the cells inherently capable of multiplying will do so indefinitely. So in other words, if like Leonard Hayflick had just known how to wash his glassware better and put the right stuff in the media, this wouldn't have happened. It was an in vitro artifact. I like to point out that letter was signed by Peyton Rouse, who won the 1966 Nobel Prize in Medicine. And one of the reasons I really like this story is, if you think about it, Rouse sarcoma virus, what Peyton Rouse won the Nobel Prize for, was really the first hint of oncogenes. That's where SARC and then other viruses like that, where RAS and MYC and things like that were discovered. So oncogenes were really, the first clue came from Peyton Rouse. And if you think about it, senescence is really the first clue to tumor suppressor genes. This actually preceded the other studies of cell fusion studies that showed benign features were dominant phenotypes. So the first clue about tumor suppressor genes really was Leonard Hayflick's work. So I like to make the point that the oncogene guys have been dumping on the tumor suppressor guys for at least 50 years. That has certainly been the case throughout my entire career until finally they put one of us tumor suppressor guys in charge of the NCI. So things are going to change. By the way, here's a photo recently. This is like from last year, maybe a year and a half ago in Santa Barbara with Leonard Hayflick, who still goes to meetings and is quite bright and asks terrific questions and is an interesting individual if you ever get to spend time with him. So the senescence theory of aging is this. I think I'd probably, that first part about the aging cancer, basic science, this is sort of a summary of 15 years of work. But it's this idea that senescent cells accumulate with aging, and in our lab, they accumulate in a way that we think is exponential. It's not linear throughout age. It's much more rapid as organisms age. And this is good for a lot of reasons. I alluded to tumor suppression, but it also plays roles in wound healing and limit of atherosclerotic plaque size and a few other important disease-related phenotypes. But such cells are also bad because they untowardly produce some signs of aging. They don't do what they used to do. So if you think about a senescent T cell, it can't really divide in response to its cognate antigen, and so it may not be useful for fighting tuberculosis or whatever it used to do in a young individual. They actually occupy key cellular niches. They cause anatomic obstruction, and they elaborate these hormones I talked about, the SASP hormones, which we think contribute to this issue of inflamma aging, this sort of chronic inflammation that's seen in aged mammals. And so that is, in a nutshell, the senescence theory of aging. And one implication of this, one of the real sort of first, I think, kind of reduction to practice discoveries along this line was a series of studies from my lab with Sean Morrison and David Skadden in 2006, where we basically showed that animals that lacked P16 were in some ways very prone to cancer, but yet they were resistant to aging. So they had this explicit trade-off that their cells in some ways functioned better. They were age-resistant, but that wasn't in general a good thing for the organism. It led to the organism getting cancer and dying young. And so when I was trying to explain this to a journalist on NPR, I said, I called that the James Dean phenomena. Live fast, die young, and leave a good-looking corpse. And you don't ever want to make a mistake like this on NPR, because then what happens is you get a lot of email that explains it is not actually the James Dean phenomena, It is attributable, it's to an older book from 1947, I think, by this guy called Knock on Any Door, and the main character says that's sort of his goal. He's a troubled youth, and then it gets turned into a pretty good movie with Humphrey Bogart as the lawyer. So anyway, that's where the title comes from. It's this idea there's an explicit trade-off between you can do things about aging, but often they'll promote cancer, and you can do things about cancer, but they may promote aging. It's a double-sided coin, and so how to work, it's a sort of box. There's no free lunch, if you will, here, and how to affect aging without causing cancer has been sort of the thing that's been interesting to my lab since then. So if this model were right, and one of the things I like about this model is it makes explicitly testable human predictions, as opposed to lots of the other sort of models of aging that have been floating around for 50 plus years. This one actually makes some predictions that we will know if they're true or not in short order. And so here are 3 of them. The first is that humans, since there's genetic heterogeneity of all human phenotypes, more or less, there ought to be heterogeneity about how people molecularly activate senescence. And if there is such heterogeneity, those people ought to age differently, if you will. And what would be a sign of aging differently? Well, I would argue one sign you could look for in existing disease data is the susceptibility to multiple age-related diseases. So you wouldn't want to just say, let's just study cirrhosis or any specific disease, because there might be some private process unique to that organ. But if you could find mutations that made people get lots of diseases in a different way, sort of a segmental progeria, if you will, or some version thereof, that would be, there ought to be such things linked to senescence, if senescence really were an important determinant of aging. And then you also ought to be able to count these senescent cells in an individual in some way and measure their physiologic age. So the in vivo burden of senescent cells ought to correlate with the age-related behavior of that individual in terms of their physiology. And lastly, if the senescent cells are actually poisoning the organism, if they're actually pathologic in some way, then getting rid of them ought to be beneficial, and that's the senolytic hypothesis, the idea that you can deplete senescent cells and make an old individual young. I have to say, for those of you who aren't sort of aging researchers, you have to understand how important that last one is, because it's very different from everything else therapeutic in aging research up until this time. So the prior paradigm has generally been prevention. You take something, green tea, exercise, sirtuin activating compounds, whatever, for 40 years, and then, at age 80, you can jog a mile further or something, right? So that is a paradigm from a drug discovery point of view is very uninteresting to pharmaceutical companies. Long studies that are prevention, large number needed to treat, that kind of thing. Whereas the treatment paradigm, you can take an aging individual and revert aging physiology, is much more like cancer therapy and things that pharmaceutical companies are much more helpful with. They do better. So I think that, in terms of interesting industry in aging, the senolytic idea has been very important. So let me go through them really quick. So set points, I think, this one is I think, no longer controversial. We actually started out to do some studies to look at human genomic loci involved in senescence and various disease-related phenotypes. And then we got scooped like nobody's ever been scooped before by like 2 million patients worth of GWAS data by people like Francis Collins and David Altshuler and others, showing that there were these recurrent hits to loci related to senescence in the human genome associated with various diseases. And that was so striking to us, we decided to download all the GWAS data that had been reported at that time and threw out all the things that were traits, so red hair and cleft palate and things like that, and restricted our analysis to actual diseases and binned all the hits of the human genome to discrete loci, 200 kb bins, but the bin size is unimportant in the analysis, and then determined significance with a permutation test. And this is what the result of that looks like. You see pretty quickly that there's one locus in the human genome, 4 contiguous dots, so that's 800 kb region, where lots of human diseases are associated by GWAS. And if you look at what those diseases are, it's things like scleroderma and type 1 diabetes and rheumatoid arthritis. And that chromosome 6 locus is famous as the MHC locus. So it was, kind of the positive control, because it was already known that many diseases mapped to that locus, especially diseases involved in autoimmunity. And then the next biggest hit in the human genome is this locus, the INCARF locus, or the CDKN2A locus, which is what encodes P16 and a related gene called ARF and another related gene called P15. And those are basically the only genes there. It's kind of a gene desert. In fact, I got in a bit of an argument with my old friend David Altshuler when he told the New York Times there were no genes there, and I was like, yeah, there's one there, and it was the one I had a Nature paper on, and you ignored it. So we had a lively back and forth about that. But in any event, and this locus is mostly associated with disease of aging. So atherosclerosis, cancer, type 2 diabetes, glaucoma, a couple more. And in fact, it's hard to think of a disease of aging that's not in some way associated with the Incarf locus, with the possible exception of Alzheimer's disease, a neurodegenerative disease. And then the other, here's the significance test here. And the only things else that are significantly associated, that have a more than expected number of hits are basically all related to either immunity and inflammation or 5P, which is where the catalytic subunit of TERT is, the telomerase enzyme. And that is all, telomere length is a major determinant of senescence in vivo, and telomere biology is really strongly related with p53 activation and senescence and aging. So I would say that by this analysis, if you think, are there loci in the human genome that are associated with lots of diseases, and those diseases appear to be linked to aging, you kind of, if you think that multi-disease association is a phenotype of aging, and it is, then you kind of identify two processes from this 2 million patients worth of GWAS data. You get senescence, these two, and you get inflammation and immunity, which are all the other hits. And you don't get anything else. You don't get IGF-1, you don't get sirtuins, you don't get reactive oxygen species. For whatever reason, those processes don't appear to correlate with age-related susceptibility to disease in large human populations. Just to make the point here about the Incarf locus and its very strong association with diseases of aging. And by the way, this is a pretty old slide now, 2012. Since then, there have been a lot of other GWASs also hitting this locus for cancers and other indications. And now, the atherosclerosis link is built into the polygenetic risk scores. This is sort of the strongest common determinant of atherosclerosis in the human genome. It's still of unclear reason exactly why. So measuring aging, let me turn to that. So anyway, I think those data establish that that the senescence machinery is linked to how we age. It is not true, though, to say that people who have hypomorphic variants, for example, live longer or something. That is not the case. So there appear to be trade-offs. The defective P16 alleles may have an advantage in some ways, in terms of some aging phenotypes, but have the cost of accelerated cancer. And that was the finding in the mice, is that, again, there's this sort of two-sided coin between aging and cancer, demonstrable in human populations. So let's talk about measuring aging. This has been a more difficult field. As I said, we've spent a long time looking for biomarkers of senescence that could be reproducibly done in a human biopsy of some sort. And telomere length was something we and others tried. That technology has some frustrating aspects to it. There's a lot of senescence that occurs that's telomere independent, so we didn't have much luck with that. We tried measuring P16 in many different cell compartments. As we noticed that P16 was about the most dynamic transcript associated with aging that we could find and senescence, and eventually hit upon this test. And the reason, and I will stipulate this is not the perfect test, but this test has the virtue of being cheap and doable in everybody pretty quickly. And we've even done this in people with end-stage HIV who have very low CD4 counts. So it's even useful in some contexts where it would seem challenging. And the idea is you get T cells from people. T cells make a lot of P16. When they do, they don't divide, and they express at least one or two molecules associated with the SASP. I think it's debatable as to whether those should really be considered senescent or not. But in any event, those cells, that's the cell that's easiest to obtain from serial biopsies in people, peripheral blood, that act as if they're senescing. And we tried measuring both protein and message, and it turns out message is better for reasons I won't go into. And then we kept a lot of serum around and DNA, and we did stuff like telomeres and cytokines like IL-6 and things like that, most of which didn't work or haven't been so useful. And then we tried to see if your at least T cell burden of senescence correlated with anything interesting. And we spent a lot of time on the assay and eventually commercialized it, as mentioned, through Healthspan Diagnostics. And now it's been done in a couple thousand people in a variety of clinical settings. And generally, they all go like this. The patient is going to have something challenging done to them. For example, they're going to have big cardiac surgery. They're going to go combination chemotherapy or radiation for cancer. They're going to have some planned toxic event. And the question is, does measuring the peripheral blood marker of aging prior to that event have any sort of predictive value? And I will say that for the most part, it's a pretty good assay. It's cheap, it's easily done, it's reproducible, it has a low failure rate. The one problem with the assay that persists and we have not been able to fix is this issue of the analyte. So we can't really get it to work on peripheral blood mononuclear cells. You have to do it on T cells, and you have to make the T cells the day of the phlebotomy. So that provides some challenges in doing the assay that are not ideal and limits what kind of retrospective studies you can do. But other than that, I think for the most part, it has been successful. And we've done a lot of stuff with it. Here's the first study we ever did, which showed that your chronologic age on this axis correlates with your peripheral blood P16 expression on this axis. And note this is a log base 2 scale. So each 1 unit increase is a doubling of the peripheral blood P16. What we think this reflects is not every T cell making twice as much P16, but rather, we believe a 10-fold increase in the peripheral blood really reflects, a 1,000-fold increase in 0.1% of the cells. So we think basically there are very few cells that make P16 in a young individual, and as you age, there's a slow but inexorable increase in the number of peripheral blood T cells that are senescent, if you will. And that's what the increase in P16 reflects. That should be provable by other sort of single-cell technologies, but to date, that's been more challenging than you would imagine. So it's a pretty good marker. You can see that one thing that's nice about it is a 10-fold increase, very easy to measure. So your grad student can get this to work like on the first try. You can see there's a big change even between people who are 30 and 40 and people who are 70 and 80. So this is a limitation of telomeres. If you look at their length, they're kind of flat in the middle lifespan, and then they drop off at the cliff at the end. Whereas this marker is more smooth throughout the lifespan. And it works out to be about a 1.5-fold increase per decade. That'll come up later. So we've used it for a couple things. One example, because the topic today is the link between cancer and aging, is measuring aging in patients getting chemotherapy. And I should say, as an oncologist, we've known for a long time that these highly toxic therapies of radiation and DNA-damaging agents like alkylating agents and topoisomerase inhibitors, they'll cure a patient. They're useful for adjuvant therapy, and I deeply believe in those therapies for the right patient. But they don't just have acute toxicity. The patients get better from the acute myelosuppression, the hair loss, the asthenia of the initial treatment, but those patients have lifelong sequelae of those therapies. And so we see that as diminished wound healing and increased risk for certain things like atherosclerosis and increased risk for things like bone marrow failure and myelodysplasia. So the idea that chemotherapy in some ways age-promoting is not surprising to oncologists, but the idea here was to actually measure it and see if we could demonstrate it. And so we did a study where we measured the molecular age using this P16 test in individuals with breast cancer prior to chemotherapy, and it was basically the chemotherapy regimen was left up to the oncologists, but it had to be at least 4 cycles of an anthracycline, adriamycin, and Cytoxan, an alkylating agent. And then we measured the molecular age at multiple time points after. And it turns out this doesn't matter. We didn't know when we initially did this trial, but you only have to wait long enough for the T cells to recover. So the T cell count goes down with chemotherapy. It recovers generally about 6 weeks after therapy. And then once it's recovered, it doesn't matter if you measure it 3 months or 6 months or 12 months. And the results are shown here. So here's patients. Each patient is a line. Here's their pre-treatment level, their post-treatment level, 33 patients. And you can see in all cases, essentially, there's a pretty substantial increase in P16 levels with chemotherapy treatment. It's sort of a mean 2-fold increase, and I remember I said it was about 1.5-fold per decade, so that's sort of like 15 years of chronologic aging, which again, I think if you asked oncologists, how much older do you think the bone marrow of my patients is with 4 cycles of anthracyclines, they'd say, 20 years. That's in terms of risk of leukemia and myelodysplasia. I don't think that's a shocking result. But it is interesting. Some patients go up a lot, some don't go up much at all. And then there's one patient you can see here actually goes down. This is an interesting patient. I like to point out, this was the youngest patient on the study. Most women with breast cancer over the age of 60 in the United States. This patient was in her 30s. So she had the highest P16 pretreatment of almost anybody. She's cured of her breast cancer and then relapses a few years later, or dies a few years later of a second primary of a different cancer, pancreatic cancer. And it turns out that she has Lee-Fraumeni syndrome. So she has a P53 heterozygous deficiency. So I know why her P16 is high pre-treatment. She had a congenital deficiency of DNA repair. P53 is one of the major guardians of the genome, or the major guardian of the genome. And haploinsufficiency is clearly phenotypically important. But I don't really know why it goes down with treatment. She's a biological outlier, but nonetheless, even included in the analysis, the statistics are highly robust. And by the way, this would have been surprising if it didn't work. We'd done this experiment in mice, and you get the same result. If you give a mouse or a rat radiation or cytotoxic chemotherapy, you get the similar increase in the burden of senescent cells just like this. So then let me briefly turn to this topic of senolysis. And this is this idea that senescent cells can be partially cleared by either naturally there's sort of clearing of senescent cells by immunologic mechanisms. And if senescent cells cause some aspects of aging, could increasing that rate of senolysis ameliorate age-related phenotypes. And I will say, one of the most remarkable things I like to point out, I've been going to aging meetings, scientific meetings a long time in my career. And the most recent Montreal Senescence meeting was unique in my time as a senescence researcher in that essentially every speaker in the senescence session, which was like 15 speakers, started with a disclosure slide about how they're working with Unity or Ocean, or there are these 7 or 8 companies that are trying senolysis through different approaches, which is very different. It is this sort of commercial interest. And some of these approaches are listed here. So you can do it in mice with genetic tricks, like this ink attack mouse that I'll show a little bit of data from. But one can also do it in any organism using small molecule inhibitors like BCL inhibitors, through an immunologic approach like a CAR T cell engineered against a senescent cell or an NK cell, for example. And I will say, in our experience, the benefits of senolysis vary by experimental system. So the details of how you do it, which molecule you use or which genetic system you use, give very different results. And in our hands, at least, the genetic models always worked better than the small molecule approaches. I think that would be hard to know that if you read the literature, but I think that's generally true statement still today. I'll show you one reagent that we are in the process of publishing is a knock-in allele where basically tomato marker, chrome, turns on under control of the P16 promoter, and it does what you expect. So the cells aren't red. These are MEFs in early culture, but as the cells are cultured, we've known for a long time that senescence is activated and P16 turns on and the cells get red, right? So it's a marker, it's an in vivo marker of senescence at the single cell level. It took us over 10 years to make this allele for a variety of reasons that I won't go into, but it appears to work. And now we've been using it for stuff like this. And so here's an in vitro experiment, but you can culture chondrocytes, the cells that make cartilage, and they will, a significant fraction will undergo senescence in vitro, and they will turn red if they're from this mouse. And then if you treat them with navitoclax, which is a potent BCL2 and BCL-XL inhibitor, you kill all the senescent cells. It's really cute, right? You don't kill the non-red cells, you're just the ones that activated senescence. We now know that senescence, and think about it, it makes sense. They're damaged cells, and yet they don't die. And the reason they don't die, it turns out, is that they potently repress apoptosis through the expression of BCL molecules. And we believe the main one now is actually BCL-XL. So this is the kind of thing what one could do with senolytic compounds or senolytic therapy. This is the experiment from Jan van Deurzen that was on the cover of the New York Times. This mouse and this mouse are littermates. And they're genetically engineered to express a toxic molecule that will lead to cell death when the animal is fed a compound. And that toxic molecule is under the control of the P16 promoter. And I would say a weakness of the senolytic field, all the mice that work use the P16 promoter, which from my point of view is not ideal. There ought to be other ways to mark senescent cells than just P16 expression. But in any event, this was sort of the first system and is the most widely used. And these littermate animals differ only in one way. They're the same age. This guy was just allowed to age, and this guy was fed the senolytic activating compound. And this guy is much healthier than this guy. And this is not merely cosmetic. You can show the animals have enhanced renal function, they have enhanced cognitive function, they have enhanced wound healing. They appear younger by every possible sense. And the senolytic compound can be started when they're pretty old. So you can wait till the mice are 20 months old, start treating with senolytics, and have reversion of age-related phenotypes. This result was kind of shocking when it first came out, but I'd say results like this have now been substantially reproduced by multiple labs using multiple different reporter systems. This also includes a lifespan advantage, which is significant, 100+ days. So they live longer. One thing that I thought was interesting, working with Judy Campisi, my lab got interested in this topic of fatigue, which is a big problem in cancer therapy. So if you ask patients who get cytotoxic chemotherapy, 5 years after they're cured, what was the worst part about chemotherapy? Some of them say the hair loss, some say the change in appetite, but many of them say just being tired for 6 months. Asthenia, chemotherapy-induced fatigue, is a limiting toxicity in many patients. And Judy did an experiment where she induced senolysis in animals with or without chemotherapy treatment. And so the results are here, and measured activity by how much they run at night. So mice run at night, and you can see that normally mice will exercise with or without senescence. If you give them doxorubicin, that'll make the animals have an increased senescence. They exercise less, and you can revert that by giving ganciclovir, which is the way to activate senescence in these mice. So senescence will restore the chemotherapy-induced fatigue in mice, and they'll hang on to a rod longer, a grasping sort of a strength assay. And the reason I thought this was so interesting is because we had the same result in people. So we did a study in breast cancer patients where we looked at P16 treatment prior to therapy. The patient then got chemotherapy of the physician's choice again. This turns out to be a mistake in trial design. I can tell you why if you're interested. And then we measured grade 3/4 toxicity, including asthenia, which is chemotherapy-induced fatigue. And we showed that while none of the hematologic toxicities correlate with pre-therapy P16, neuropathy might. It doesn't happen frequently enough to say it's more common in people with high P16, but asthenia does. So you can show the people who have asthenia have a higher P16 than people who don't. It's a log scale, so a half log is like 40% or something. And you can show if you bend the patients by quartiles, the patients with the highest P16 are 10-fold more or 9-fold more likely to have asthenia than people with low P16. So the point is that having a lot of senescent cells is associated with asthenia, particularly in the setting of chemotherapy treatment. I don't think this should be surprising, actually. If you think about what asthenia might be or what causes fatigue in chemotherapy-induced patients, senescent cells make IL-6, IL-1 beta, IL-8, the exact same sort of hormonal milieu you make when you get a cold. And so, sort of the fatigue associated with viral infection is not maybe dissimilar in some ways from the fatigue associated with a large burden of senescent cells. So I've come to say that getting old now is like having a cold all the time, which I believe more and more every day. So let me just sum up the state of the art of senescence going on right now. So as I said, there have been multiple labs now starting to get these senolytic mice or these senolytic molecules to work. And so it looks, as I said, reproducible. The other thing I'll say that I find really encouraging is that, for about a decade, a lot of our progress in senescence was really descriptive. It's in this compartment, it's not in this compartment. If you cree out P16, you get this phenotype, not that phenotype. But recently, I think there have been a number of really important mechanistic discoveries about what makes a senescent cell, how the signaling module works, the chromatin markers of senescence. It's really been nice to see. And for example, this unexpected finding, I think, from Dao-Hung Zhou that it's really BCL-XL is a survival molecule. I don't think anybody saw that coming, but appears to be true. And then there are actually, as I said, a number of clinical trials. They're not just companies trying to do this, but they're actually studies in people. Two that are worth mentioning. This is a study using this combination is thought to be senolytic, and the idea is to try and repair damaged kidneys, so giving it to patients with chronic renal failure. And this is the Unity compound, and I think Unity thought that giving systemic senolytic would be a challenging endeavor, so they decided they'd start with intra-articular injections in joints. So the idea is that old joints have senescent chondrocytes, I already showed that, and the idea is by depleting senescent cells in a joint, you might be able to reduce age-associated osteoarthritis, which is a big market. So now let me turn to a different kind of iatrogenic aging, and that is this related to stem cell dysfunction. And so hematopoietic stem cells, their aging is well studied, and it is not related to senescence. Hematopoietic stem cells divide just fine in old and young organisms, but what they choose, they change in their epigenetic state. Their ability to make blood wanes. So there's this proliferative, how many blood cells can a given stem cell make? That goes down. And there's also this bias. So they stop making stop making lymphoid cells and prefer myeloid cells. And this can be induced by chemotherapy. So oncologists are familiar with this phenomenon where if patients are serially treated with chemotherapy, at some point, even if their cancer is still responding, they will develop this sort of stem cell exhaustion business where their platelets don't recover, their red cells don't recover. And so even though the chemotherapy is still working, the bone marrow toxicity becomes limiting. And there's a lot of genetic evidence, RNA expression and other kinds of phenotypic studies that show that hematopoietic stem cells that are induced by aging, by serial transplant, or by serial chemotherapy treatment look biologically similar to physiologically old stem cells. We think that physiologic aging and chemotherapy-induced aging is similar for hematopoietic stem cells. And this study is an effort to prevent that from happening with chemotherapy, and it takes advantage of a trick with the cell cycle. And so in 2005, this is sort of how we thought the cell cycle worked. This model is wrong in sort of every possible way it could be wrong. And the model basically was refined in sort of about 2006 by a bunch of knockout mice. So Phil Caldus and Mariana Barbacid and others made a bunch of animals, Peter Szynski, and showed that there wasn't this linear, this series of phosphorylation events that regulated entry to the cell cycle, but rather it was sort of a parallel circuit. And some cells would use CDK4 and some would use CDK2. Some cells could only use one, some cells could use any of the three. One of the ways you knew this model was wrong is if it were right, every proliferating cell should need CDK2 activity. And that turns out to be untrue. So if you knock out CDK2, the mouse is basically fine. So it's really a stunningly negative result if you think about it in retrospect. But one of the reasons why this is relevant today is it meant that when this was the idea, pharmaceutical companies were avoiding the cyclin-dependent kinases for cancer because they thought they would be very toxic. But when this became the model, it became clear that you could drug maybe CDK4 and not affect CDK2, and that would be less toxic. And so those sorts of molecules came into existence. So we got our hands on one of these a long time ago. At this time, this drug was called something 991. It's a Pfizer molecule. It's now called palbociclib. And this drug is FDA approved today for hormone receptor breast cancer. But we tested it just to ask the question, given this new model of the cell cycle, what tissues in an animal are CDK4/6 dependent, which the enzyme, this compound inhibits, and what cells proliferate in a CDK4/6 independent way in an adult mouse? And so we showed like beta cells in the islet are highly dependent, and that's interesting and related to, I think, the association of CDK2 in 2A with diabetes. And then, there's cells in the pituitary that are CDK4/6 dependent. Basically, all lymphocytes, B and T cells, turn out to be CDK4/6 dependent. But most dividing cells in the gut and the bone marrow are not. They divide just fine. They can use CDK2. They don't need CDK4/6. And so you could give these drugs to mice for months without any real toxicity. You get modest myelosuppression, but for the most part, they tolerate it pretty well. And this has now been true in humans as well. So women with breast cancer have been on palbociclib for years with minimal toxicity. It's pretty well tolerated. And so this meant that you could modulate the proliferation of some cells with minimal toxicity, and that means you could get rid of cells you didn't want, like a breast cancer cell. You could prevent them from dividing, and that would be useful, and that's what Pfizer had in mind for palbociclib. But you could also protect cells that you wanted to keep around from a cell cycle-specific insult if they were CDK4 regulated. And that became this concept of pharmacological quiescence. And so here's sort of some cells you might want to get rid of. Neoplasms has gotten the most focus, but I think the use of CDK4/6 inhibitors for immunity, autoimmune disease, is an interesting idea that has not been taken up to date. And then you might want to modulate the proliferation of macrophages in some setting, which is also CDK4 dependent. And here's some cells you might want to protect. I won't talk about the kidney today, but it's a fascinating story. The kidney is a deeply quiescent organ that doesn't divide unless it's injured. And then the epithelium of the kidney rapidly goes into cycle, and that cycling is completely CDK4/6 dependent. So if there's a period where renal proliferation is unwanted, you can stop it from happening with a small molecule CDK4/6 inhibitor. But I'll talk mostly today about hematopoietic stem cells and progenitor cells. So we tried to use Pfizer's drug and the other compounds available and couldn't, and eventually just ended up making our own molecule. And this was the story of how G1 Therapeutics came about. And the compound that company made was called trilaciclib. It's shown here. It looks like ATP. It is a type 1 kinase inhibitor. It works the way you would expect, inhibiting the proliferation of RB competent cells, but not RB null cells. This compound was designed to be given intravenously because it was going to be used for chemoprotection, and one would want to control the exposure of the drug at the time of the chemotherapy administration. Has a relatively short half-life, so most of the CDK4/6 inhibitors that were developed for clinical use were designed to be taken once a day orally, right? So that it was your cancer pill, and you take it every day, and taking it one time a day would be easy to remember. But this compound the intent was to be given IV at the same time as the chemotherapy dose, and so it has a much shorter half-life. Because the idea is you want to protect the stem cells, and then once the chemotherapy is gone, you want the drug to go away so that the stem cells can start dividing again. Here's just a summary of the existing CDK4/6 inhibitors. Trilaciclib I mentioned, palbociclib is approved, ribociclib is approved, abemaciclib is approved, liriciclib is in clinical trials. They're, for the most part, they're similar. The one that's different is abemaciclib. It is a staurosporin derivative. It is a planar molecule that is much more promiscuous in its ability to hit kinases. But the other ones are quite similar in their at least activity against CDK4 versus CDK2. And the important thing to note here is that we found as we were trying to do this, we didn't really know what kind of CDK inhibitor would be best for chemoprotection. And the answer is you want one that inhibits 4 and 6 really well and doesn't hit 2 at all. So you need like at least 1,000-fold difference in the potency. And that turns out to be very important for reasons I could go into if interested. But so those are the molecules that went forward. I'll briefly remind you that there's this hierarchy of hematopoiesis. I've already said that these cells here, the lymphocytes, are dependent on CDK4. Peter Szynski actually discovered that these cells up here are dependent on CDK4. And then pretty much all this stuff over here is not dependent on CDK4/6. So that's why you can give patients an inhibitor and they'll continue to make blood and platelets for quite a while. And so here's sort of a way of showing that. If you give an animal a dose of trilaciclib, you inhibit proliferation at very low doses of hematopoietic stem cells and lymphocytes, but not of like erythroid progenitors. And here's what happens when you give a single dose of the drug. The animals, the hematopoietic stem cells stop dividing for about a day, and then they start dividing again. And notice they always proliferate extra. They hyperproliferate as the drug goes away to compensate for the period they were out of cycle. And so, here's the sort of thing you need to know is that toxicities like radiation are cell cycle dependent. And this has been known since like the 1950s. So the idea is if you damage a cell early in G1, it's pretty resistant. And if you damage a cell late in G1, right as it is about to traverse into S phase, it's quite sensitive. So the notion is that if you could take these hematopoietic progenitors and hold them in G1 a bit longer, you would make them resistant to something like chemotherapy, to a DNA damaging molecule. So it works in mice. Here's untreated animals in the gray. Here's animals that get 5-FU, a way of inducing cytotoxic damage. And then here are the blood counts of animals that get 5-FU and a CDK4 inhibitor at the same time. And so you see you protect neutrophils, lymphocytes, red blood cells, and platelets with a single dose of the drug. It's quadrilineage protection. So these studies have now been done in people. Just to remind you that this is important, hematologic toxicity is a really big problem, particularly in diseases that use these myelosuppressive drugs, which do work. They are not going away in oncology anytime soon. Adriamycin, platinum, etoposide are very useful drugs in certain patients, but they are hard to give. Most patients need breaks in intensity. They have to have their doses reduced, and myelosuppression is generally the dose-limiting toxicity of these agents. And so the studies of trilaciclib in people all pretty much look like this. There's trilaciclib is given at the same time as a chemotherapy regimen, which can be a 1 or 2 or 3 day regimen, and it's done every 3 weeks on a cyclic fashion. 3 of these studies are in small cell lung cancer, and 1 of these is in triple negative breast cancer. And the reason those diseases were picked is because those are cancers where RB deficiency tends to be common, and therefore the CDK4 inhibitor won't protect the tumor. The tumor will divide, but the bone marrow will stop dividing when you give someone a CDK4 inhibitor. And the endpoints are generally hematologic toxicity. Do you ameliorate myelosuppression? And the answer is it works. So I've aggregated recent data reported for 2 different small cell trials. These are both using the regimen etoposide platinum, 180 patients in a randomized two randomized phase 2 trials. And there's pretty good reduction of all these hematologic phenotypes. So there's less neutropenia, there's less periods of neutropenia, there's less GCSF usage, there's less blood transfusions, there's less platelet problems, and there's greater dose intensity, so less need for dose reduction, and it's highly significant for almost all the phenotypes except grade 3/4 anemia. So, but the survival data are still cooking. It's unclear that protecting the patient's bone marrow helps them in any way. We believe it does. If you can give more chemotherapy, that should work better, but that remains to be seen. The other points that we don't know if there are other tissues besides the bone marrow that divide in a CDK4 dependent way. And so are you protecting them? For example, the kidney I mentioned, or hair follicles, alopecia, for example. So are those things protected by this approach as well, or is it just the bone marrow? And the last thing we don't know is if this is all about the acute toxicity of myelosuppression, but what I got into this whole business in the first place is I was trying to prevent aging, the long-term sequelae. And so the notion is that by protecting the progenitor cells, you would also protect the stem cells. And so the idea here is that by protecting the stem cells, that would have long-term benefit for the organism. That's hard to test in people. We won't know that for a very long time, but it is very doable in mice. So here we caused hematopoietic aging of the stem cells by serially treating animals with 5-FU, and we either did or did not give them a CDK4 inhibitor at the same time. And then after 4 rounds of that, you take the stem cells out and you transplant them into an irradiated mouse, and you look at the ability of the mice to make blood. And very quickly, the results are shown here. Cells that don't get any 5-FU at all make blood really well, so they make 50% of the blood because there are also 50% competitors transplanted. Animals that get 5-FU don't do so well for any lineage, and animals that get 5-FU with a CDK4 inhibitor are rescued in all lineages. Again, this is a log scale on these cases here. So the ability of hematopoietic stem cells to make blood is protected by concomitant CDK4/6 inhibition. So we believe this is evidence that we would protect a serially treated human from chemotherapy-induced iatrogenic aging of the hematopoietic stem cells. Another thing that we noticed in the mice is that if you give them 5-FU, these are animals that are not transplanted, and you just look at their ability to make lymphocytes 100 days after that, it goes down. So untreated mice make lymphocytes really well. 5-FU treated mice lose their ability to make lymphocytes and have this myeloid skewing in the peripheral blood, and you can see that is also rescued by concomitant administration. So we show that the stem cells from animals that receive a CDK4 inhibitor make more blood, and they don't have the myeloid bias. So the two sort of hallmark features of hematopoietic stem cell aging are both rescued by administration of CDK4 inhibitor. So in summary, cytotoxic chemotherapy cures cancer. That's really important to say, but it also causes some aspects of aging, we believe, by leading to accumulation of senescent cells. These senescent cells appear to drive some of the toxicity of cancer chemotherapy. Cytotoxic chemotherapy also induces the stem cell exhaustion, in part by depleting hematopoietic progenitor cells, and therefore has a toxicity of hematopoietic stem cells. And then pharmacological quiescence at the time of cytotoxic chemotherapy appears to protect from stem cell aging, at least in mice. Here are the people that did the work, many former collaborators at UNC, these various companies. I'd like to point out Artie Huria. Artie was a great colleague. She was very instrumental in the studies of P16 in humans. So those cohorts I talked about, women with breast cancer, a lot of those studies were done in collaboration with Artie's MyCarg consortium. Artie died about 2 weeks ago in a car accident. This is a particular tragedy because it's a beautiful person who was a mother, a wife, a scientist, and a doctor. But it's a particular tragedy for geriatric oncology because she was really one of the few geriatric oncologists out there who got this relationship, who understood that the biology of aging would have clinical consequences for people. And she was very interested in making this translation. As I go nationally, and many places are sort of trying to start a geriatric oncology program, they ask me, are there people like Artie and Hymus and a few others who are interested in biology and translation in geriatric oncology? And there's a real shortage. I usually tell them to try and train their own. So this is really devastating for the field because Artie was a particularly good leader and a real a forceful advocate for this topic, and will be sorely missed. So that's my talk today. I'd be happy to take any questions. Thank you for the time.
Thanks, Ned. We do have time for questions, and the microphones are available so that the video listeners can also hear them. We'll start right over here.
Well, congratulations. So you think I could get rid of those cells. So one of the areas where your approach might be interesting in the formation of the organoids. As people are growing the cells, they have this cue of making three-dimensional matrix with the matrigel. So after a while, they keep growing till 180, 200. Suddenly, the system collapses. So your work reminds us that maybe some of the drugs you mentioned as the senolytics or other variants might be useful. So what do you think is the best one they should try?
Yeah, I think there's something interesting thing about the organoid topic, and also the precursor to the organoid technology was the ROC1 inhibitor, the ROC inhibitor grown cultures. It's really interesting that, so some of the cells in those cultures, in cancer at least, are cancer. And they've inactivated senescence. That's a hallmark feature of malignant cells is they kill off P16, they kill off P53, RB, stuff like that. That's why I got interested in the field in the first place. But also those cultures also involve the long-term propagation of non-malignant cells. And it works pretty well in these culture systems for some cell types. And an interesting question is, why don't they activate senescence? Because if you just throw cells in a dish the way Leonard Hayflick did in the 50s, they rapidly turn on senescence, either very acutely because of sort of reasons related to oxygen radicals in the media, or over a long term in a telomere-based way. And yet that has been less limiting in some of these systems. And I still don't know why. I've been asking people who work on this kind of system for a while this sort of question. The ability to test senolytics in those kinds of systems is a really interesting idea. I'm unaware of anyone doing that today, but I bet you someone is. Because sort of every high-throughput screen for small molecules that will get at this question of senolysis, there's somebody doing it. It's really amazing how the influx of individuals, you sort of any kind of high-throughput screen, cell-based screen that will get at the senescence question, I've seen people doing. So I think it's a good idea, but I'm not aware of an effort, but I wouldn't be surprised if there was one.
Great.
Thank you.
Over here.
Hi. I have a question about the role of accumulation of senescent cells in causing aging and the benefit of senolysis or clearance of those cells. So you showed clearly that the senescent cells can make cytokines like IL-1 beta and IL-6 and so forth that can cause some effects of aging and inflammation. But I'm wondering whether their presence also inhibits the development of new younger cells that might be more functional. For example, we know in the immune system from adoptive cell therapy studies that lymphodepletion is necessary before transfer of adoptive T cells to allow them to get established. And it used to be called making space, but now it's believed that the existing T cells are competing and consuming cytokines like IL-7 and IL-15 and so forth. So I wonder if something similar is happening with the senescent cells, that they, just their very presence prevents the generation of younger, more effective cells.
Yeah, the issue of why senescent cells are toxic has been very hard to prove. I'd say there were sort of initially two camps. There was the senescent cells are bad because they don't do what they should do. They don't divide. And one of the leading proponents of that camp was me. And then there was the senescent cells are bad because they make stuff that makes everybody else in the neighborhood unhappy, the paracrine senescence effects. And I'd say the leading proponent of that through the years has been Judy Campisi. And I think we're both right. I think it turns out that both of those things are bad, and neither of those lists were complete. That a third possibility is that senescent cells are bad because they occupy niches. That's clearly true in what you alluded to, and I think there's some good examples of that now. And then the fourth is that senescent cells are bad because they get in the way of, they block stuff, and that's probably how senescence contributes to the size of an atherosclerotic plaque, for example. And that may be, from a public health point of view, one of the most important pathogenic mechanisms of senescent cells. So I think our list was too black and white and too small. But we haven't done a good job of sorting out those four possibilities on a cell-by-cell, tissue-by-tissue basis. So I can't really say, like in lymphocytes, if senescence occurs, is it bad for the organism because it's occupying a niche, or because the cell doesn't divide, or because it makes SAS hormones, or all three? So those experiments turn out to be very difficult. And I think you get different results in different tissues, and probably even among different subsets of lymphocytes, you get subtly different results. So they are going to require weedy, physiologic, dedicated experiments to sort them. But I'm glad that people are starting to think about this, and we're seeing efforts to test that. And so I think that's an important development. One other thing I will say about that is that it is clear that senescent cells are, well, there's a piece of unpublished data that I think is really interesting at this issue. So in cartilage, the thinking's been for many years that chondrocytes don't divide much. And they certainly divide great in culture. They will divide really, really well, even from an old organism. But the demonstration of the proliferation in cartilage in old organisms has been hard to show after development has ended. But now there have been a few reports that if you deplete the senescent cells first, the remaining good chondrocytes can divide. So it gets to this idea that the senescent cells that are there, which may only be 3% or 5% of the cells or something, they're rare, but they're bad because they cause this local poisoning of the milieu.
Thanks. Time for a couple more quick questions. I will continue over here on this side.
You have an assay for the P16 Inc4A. However, there already is an assay, isn't there, for the methylation?
Yeah.
How does that compare to what you're doing, and why would you not choose to do that instead of go through all the problem of developing a new system of determining aging in cells?
Well, first of all, the DNA methylation stuff is much newer. We've been working on this a lot longer. The DNA methylation thing is really interesting. So the R squared between methylation clock and a chronologic age is like 0.9 something. I mean, a very strong correlation. In fact, you might argue that's a bug, not a feature, because we're not really interested in knowing someone's chronologic age. The driver's license test is quite good at that. What we're really interested in is knowing their physiologic age and what portion of physiologic age, what should the R squared between physiologic and chronologic age be? Probably not 0.9. But I think a reasonable point to make about the methylation clocks is the reason they're so good at checking age is because they were trained on chronologic age. But what if you trained them on physiologic age? They might be very good at that too, which is still an unaddressed question. So I would say right now, and NIA convened a biomarkers of aging conference like a year ago that I went to. There's we came up with like 70 different biomarkers of aging. I'd say there are kind of maybe 3 or 4 that are really ready for large human trials. There's the P16 test, there are various telomere assays, and there are the methylation assays. And all of them have pros and cons. And I would say in the one study that I know where people have really compared all 3, they all correlate with each other to some extent better than they correlate with chronologic age. So I like that, the idea that there's sort of a physiologic age that's measured by these biomarkers that all measure different things, and that's not solely captured by chronologic age. If all you're doing is having something related to chronologic age, it's not very useful. Right.
Over here.
Thanks, Ned. That was very exciting as usual. You showed a very good experiment where you measure senescence before giving chemotherapy, and you see that senescence cells increase. I was just wondering if you think there will be any predictive value, say, on taking those cells, not just measuring senescence, but inducing senescence. You're going to treat these patients with a chemotherapy.
Yeah.
So would it be a value to treat the cells with the same chemotherapy agent to see how they're going to react?
You mean as a predictive marker?
Yeah.
Yeah. That'd be hard to do. I mean, the problem is, one of the reasons why my lab kind of got away from cell culture and moved solely into mice and people is that the act of culturing a cell so profoundly perturbs senescence that it almost becomes more about the details of how you culture the cells. Does it activate it quickly or slowly or not at all? And different donors can give different results. It's really hard to do. So I think the ability to take cells out of a person and measure their proclivity to undergo senescence is challenging. I will say that question, not so much for chemotherapy response, but is of great interest to the diabetes community. Because the SNP associated with type 2 diabetes is very strong. It's maybe Francis would know better than me, but it's like the 3rd highest on the GWAS hit.
I think that's right.
Yeah, and nobody knows why. And my lab showed it's not, the P16 level in the peripheral blood T cells is not associated with the diabetes SNP. It is associated with the atherosclerosis SNP, but not the diabetes SNP. So we never could figure out why. But people who work on diabetes always made the point to us that most of those GWAS hits probably work in the beta cell itself. And so maybe that's a regulatory event of CDKN2A in the beta cell. But it's hard to get thousands of primary beta cells from different people for obvious reasons. So there was this interest by people like Chris Newgard and the real diabetologists of trying to get islets from small numbers of individuals, like 40 or 50, and measure their proclivity to undergo senescence with or without that SNP. I think there actually was a paper from Andy Stewart that was informative on this topic, where they sort of looked at the SNP and its relation to glucose clamping and got a result that I thought was informative. So it is, I think most people believe that SNP works by affecting beta cell proliferation, but the data are still not as clear because culturing beta cells is really hard.
And that'll be interesting to test when you can differentiate iPS cells into beta cells. Which
then you can do it.
You have multiple different genotypes and you can see what happens. Or you can even CRISPR-Cas edit a particular cell line and just change that one thing and see what happens. Yeah, last question.
Hi, great talk. I really enjoyed it. My question is about, is there any diurnal aspect to senescence and clearance as relates to potentially lack of sleep and levels of senescence and related to macrophage and the garbage cans that they are cleaning up nightly as they do? And so is there anything about that?
Yeah, I get a lot of these questions, and I
from sleep-deprived people, probably.
But I get asked about diet and smoking, and I will say we showed a very reproducible, strong finding is smoking behavior and the P16 marker. You actually have to control for it. It's so strong. So smoking is a very potent way to activate P16 in your T cells if you want to do that. But the diet stuff, we never did very well because we didn't, know how to ask the right questions. Exercise turned out to work much better than we expected, and I think if we'd known it was going to work so well, we would have asked more exercise questions. But then you get into, multiple behaviors, and my response has always been, We weren't really the right lab. The sleep experiments are hard. They're a lot, even in rodents, they can be confounded by a bunch of things like the animal changer coming in the middle of the night. So, I've tried to make these reagents. We've made 10 different engineered alleles of P16, reporter alleles, and conditionally ablatable alleles and whatnot. And I've tried to make them available to the community to study this. But I think particularly the biology of sleep and DNA repair is pretty compelling now. And then the linkage of DNA repair and senescence is very strong. We have not been able to find something that induces senescence that doesn't break DNA. To my great disappointment, we've always been looking for gerontogens that accelerate aging or at least accelerate senescence without causing DNA damage, and never really found anything. So I think it's a very good question. If sleep patterns are associated with proclivity to activate senescence, I suspect they would be based on what I know about sleep, but we never did it.
Well, great discussion and wonderful presentation. We can gather in the medical library if you want to ask more questions or have some refreshments, but please let's thank Dr. Sharpless again. Nicely done.